The overall goal of this procedure is to operate a miniaturized flow cytometer on board a reduced gravity parabolic flight using components, preparation, and in-flight procedures potentially adaptable to other setups. This is accomplished by first carefully selecting off-the-shelf and custom fabricated components for ease of use and safety in reduced gravity. The second step is to assemble components within a parabolic flight test rig containing additional elements for containment, viewing, automation, and facilitation of multiple demonstrations. Next, the team prepares for successful in-flight experimentation through meticulous planning, protocol development, and training. The final step is multi-component demonstrations in flight. Ultimately, parabolic flight testing is used to showcase the potential space applications of technology and to identify the effects of weightlessness, gravity changes, and vibration on performance.
Although this method can be applied to flow cytometry and related technology, it can also be applied in parts to other types of in vitro diagnostic testing in reduced gravity, particularly anything with multiple demonstrations or trigger procedures.
So we decided to do this video for Jove on parabolic flight testing because when we were preparing for our parabolic flights with NASA, essentially there weren't that many videos or even literature that described how to actually best prepare for the experiments. We actually had to try very, very hard to talk to the right people at various locations within NASA to dig up that information. Here in this case, we want to be able to share this information for the readership of Jove so that they can also prepare for these flights appropriately.
Constructing a simple flow cytometry system for use in reduced gravity conditions.